Thank you for the introduction, Samantha. Well, I decided the title of my talk, you can see there, is how to detect unexpected features and physical processes in single molecule data. I wanted to put first, you know, the acknowledgements of to the people who've actually done the work, and in particular Mario Lemmer and Anton Vladica, who, as I said, had done all the work that I'm showing you today, and also collaborate at Bangor University in neuroscience, Katja Koinicheva, who has in many ways kicked off a lot of the interest in this field. So, you know, so notice that sort of some interdisciplinary component there. Now, the research topics that we work on in the group span a wider range of areas. So it goes from a single molecule biosensing using nanopores and nanopipettes to using well, tunneling detector, quantum mechanical tunneling detector for molecule detection, and also single molecule charge transport and thermoelectrics work. What ties a lot of these activities together is that we are dealing with a large amount of data, right? Often one-dimensional, say current time or current distance data, and that in many cases the data are the data sets are large and complex in a sense that we also don't necessarily know what to expect from the data. So there may be a hypothesis, but at the same time, we may want to be unprejudiced when it comes to analyzing these data, right? And just to illustrate this with examples, so what you see here is a sort of a toy representation of a nanopore sensor. It's basically a liquid-filled cell, yeah, and I hope you can see my pointer, a liquid-filled cell which is separated by a thin membrane, and that thin membrane has got a single nanopore, a nanometer scale pore, which allows liquid and ion and biopolymer transport from one compartment into the other. Now, you've also got two electrodes shown on the left and the right, and then when you apply a voltage, you can induce an electric field in the cell, and that electric field is focused around the nanopore. Yeah, and I can go into more detail as to why this is, but for the purpose of this introduction, I'd like to focus on other things here, namely that when DNA, which is normally negatively charged, gets close to the nanopore, it feels the electric field is then pulled through the nanopore from the left compartment to the right compartment. Now, if the nanopore is small enough, and typically with a nanometer-sized pore that's the case, it would constitute the largest source of electric resistance in the cell. And that means when an individual piece of DNA travels through this nanoscale pore, it actually changes the overall current that we're measuring across the cell. Yeah, now this current depends on the conductivity of the electrolyte, it depends on the applied bias voltage, but and it also of course, depends on the size of the nanopore. A bigger pore will produce a bigger current than a smaller pore, and so on. But importantly, depending on the shape of the DNA as it travels through the pore, we actually see different types of events. So typically, you know, we'd record a current as a signal as a function of time. If there's no DNA in the pore, we just see some what we call open pore current. And then as individual pieces of DNA translocate through the pore, we see individual dips, yeah, and one here, one there. And again, these are single molecule events, typically in the way we perform these experiments. And if the DNA moves through the channel in a linear configuration, right, as shown at the top right there, then the current time modulation is simply a rectangular pulse. If the DNA enters the pore in a somewhat folded configuration, I'll sort of try to highlight or show this here, then actually you get an initial excursion in the current time event, and then it sort of reverts back to a current level that's you know, normal of a linear piece of DNA. Equally, if you have DNA with knots, for example, for every knot you get a sub-event, a spike on top of the rectangular pulse, or you have bound proteins. Again, you produce sub-events here, which are characteristic of the physical dimension of the bound protein. And you can see that this could also have applications in sensing, for example, but again, that's not the topic of my talk today. The important thing here is that event shapes vary, right? And they reflect the biophysical properties and shape of the DNA, or more generally, the analyte. I'm giving you two more examples, but I needed to introduce a technique first in case you're not familiar with it, and that's scanning tunneling microscopy. Now, a scanning tunneling microscope is in principle a very simple device. It's composed of a sharp tip, a conductive sort of needle, if you like, made out of tungsten or platinum iridium, and that hovers above a conductive substrate. Again, that could be a metal surface or a semiconductor, for example. Now, the distance between the tip of the STM and the substrate is really small. So typically in 1, 2, certainly smaller than 3 nanometers. Yeah, and if it's that close, then electronic wave functions in the tip and the substrate start to overlap, and you get electron exchange between the tip and the substrate. When you then apply a bias voltage, you actually get a net current. And because that current depends on wave function overlap, effectively, it's very, very distance dependent. Yeah? It's a technique that's very versatile in the sense that it works in vacuum, in air, and even in solution environments. Right? And these are two examples here of a gold 111 surface recorded in acetate buffer. Yeah, and you can clearly see individual gold atoms here. Notice the scale bar. This is 2.5 nanometers down here. So every individual blob is a gold atom here. So really high resolution you can get with this technique. And equally on the right, you see this is a gold 111 surface, but modified and in particular with the low density hexane thiol layer. And you can kind of see these sort of striped domains here. When you zoom in, you can see submolecular structure, right? So really high resolution technique for imaging in various different environments. You can also use the technique for spectroscopy, for example, current time spectroscopy. Now, as I mentioned, the current is due to quantum mechanical tunneling. It's not an ion current as in the nanopore device, but it's a quantum mechanical current that depends on the electronic structure of the tunneling junction. And the idea of recognition tunneling or sequencing by tunneling is that depending on, let's say, which DNA base sits in this, oops, lost my pointer, which DNA base sits in between these two electrodes here, you get different quantum mechanical tunneling signatures. Now, that's at least the idea. And this is, I should say, this is taken from Stuart Lindsay's group, published a few years ago, in this case on individual deoxymonophosphate bases. And you can see here from top to bottom, just from a brief inspection, you see that the different building blocks in the DNA actually produce different current time characteristics. Yeah. The TMP didn't actually produce a signal here, but the other ones do. But the most important outcome in the context of this talk is that actually individual events are no longer just a single rectangular pulse, for example, but they have complex substructure, right? They are bursts of individual spikes, which people believe have to do with hydrogen bond formation or bonding interaction between the nucleotide and the electrodes. Yeah. So in other words, events and their characteristics may be highly complex. Yeah, it's not just a pulse or you know, with a pulse with substructure. It can actually have some quite complex temporal patterns. Now, you can also use it in a different way, use it in the context of current distance spectroscopy. What the experiment here looks slightly different in a sense that, you know, you put a molecule on the surface or a submonolayer of this molecule onto the surface, and then you approach the surface with the STM tip. At first, typically, we actually crash the tip into the surface to have a point of reference, and then we measure the current as we pull the tip away, and under some certain, with a certain probability, a molecule on the surface will start to bridge the tip substrate gap until it's first picked up and then stretched. Eventually, it ruptures. right? And this process can be repeated thousands of times. It can be automated fairly easily, right? So you get thousands of traces, current distance tracings in the way I've shown them here in this illustration. So again, at the beginning, we have tip substrate contact, right? So we may get a fairly constant current. Then we pull back the tip, and because of the quantum mechanical nature of the process, we get an exponential decay, right? Then we've got the formation of the molecular bridge, which eventually ruptures, right? And then the current drops, and eventually we basically can extract the current that flows through an individual molecule from this step height here, from this plateau height. And indeed, we can often also extract the distance, the molecular length at the point when this junction ruptures. Now, in reality, we record, as I said, many of these traces, and then analyze them in either one-dimensional or two-dimensional histograms. Ignore the molecular detail here for now. I'll come back to that later. But just to show you, on the right-hand side, we've got a 1D histogram of the conductance. Yeah, and you can see here there's a conductance peak, which turns out to belong to the molecule. Yeah, so that's a peak for the molecular conductance. Here we've got peaks for the point-point contact, which is also interesting, but from the molecular point of view, this is the signal of interest. And here on the left, we've got the 2D histogram, which also takes into account the distance axis here. Yeah, so a red color here means a high density of data points. Blue means low density. So you get this overall plateau shape here that corresponds to the simple idea of, the junction stretching and then rupturing. You also notice there's significant variance in the data. Yeah, notice this is a logarithmic scale, and even within this plateau feature, we have a difference of perhaps a factor of 10 between, the different plateau heights. Yeah, so there's significant variance in the conductance, but also in the break-off characteristics here. And of course, well, while plateau shapes are observed following our simple model, they are not necessarily the only features that we see. And what you see here is taken from a different experiment on octane dithiol. Yeah, and the coloring is essentially a human attempt to group these traces according to similarity. And you can see here that this is not just plateau shapes. There's a lot of non-trivial behavior, sometimes dynamic behavior, involve switching, telegraphic noise. So reality as often is a lot more complicated than simply the toy model that we came up with. Now, what that means from an analysis point of view, especially when molecules are new molecular systems, is that we don't want to make any assumptions at the beginning of the analysis of what a molecular signature look like, because otherwise we may introduce user or confirmation bias. Let's say we assume that there should be a plateau shape, so we'll find a plateau shape in a large data set that is very, has significant variance, right? So we're just finding what we're looking for, right? That can, of course, lead to information loss if there's other types of behavior that we might not expect and therefore not look for it. And then we also might miss complexity in the data set. There might be subpopulations in there which are physically meaningful, but we simply miss, right? And these are all things that we need to be mindful of when we analyze data sets that are too large to go through, by hand. And again, that again complicates matters in terms of user bias and so on. So which method can we use to explore data sets like these? And again, this could be in the context of a sensing application, but it could also be for materials characterization, for example. And it turns out that dimensionality reduction techniques are really a useful tool in this context. And if you have had exposure to this before, you know, of course, that there are a range of different dimensionality reduction techniques, some of which have been used in the context of single molecule charge transport studies. And of course, there is principal component analysis, the archetypical dimensionality reduction technique. But there are others, MPVC, so multi-parameter vector-based classification, is one that we've developed a few years ago. There are deep convolutional neural networks, autoencoders, TSNE, and other techniques as well. And I will today talk about two of them. namely MPVC and a sort of a slightly more sophisticated image recognition-based technique. Now, when you think about data analysis and dimensionality reduction, it's quite useful to break this down into a sort of more generic flowchart, because that's common to essentially all of them. Now, starting off on the top left there with the measured data. So we might have 2000 data points per measured trace. And from a data analysis point of view, that would be a 2000 dimensional data set. Right? We apply dimensionality reduction, which could be any of the techniques or that I've mentioned on the previous slide. But what that does is that it then produces a reduced number of parameters, maybe only 2 or 3 out of these 2000, that then help you to differentiate different behavior in the data. And that means that, let's say you've got 2 parameters characterizing each trace. You can then produce a scatter plot where each point corresponds to a trace that initially had 2000 data points. Yeah, and so with the scatter plot, you can then use clustering methods and extract groups of data points that are similar, right, or close together in the scatter plot. By implication, that means they are similar in the original data trace, and then start to analyze the data going forward. And conceptually, that has an advantage also because the hypothesis that you use to analyze the data enters the sort of flow of events fairly late. And again, if you look at this conceptual flow chart going from data recording of large data sets to dimensionality reduction, reduced space representation, clustering, and so on, the hypothesis that you use to then analyze and interpret the result actually enters at the end here, which is different from a situation where you look for a particular feature, right, where basically the hypothesis would enter up here. Yeah, so you reduce the risk of user bias and confirmation bias when analyzing the data. Now, coming to the first method, vector-based or multiparametric vector-based classification, MPVC. Now, in the interest of time, I will not go into a great level of detail, but just highlight how this method helps us to extract information from data that at the point when we recorded them was unexpected. Yeah, and the theoretical basis for this was published in this Nature.com article in 2016. Yeah, on the top left here, that's based on simulated data and experimental data. We've since then applied this to a range of different experimental systems. And it works quite nicely in many cases. Now, as I said, I won't go into great detail here, but the basis of MPVC is that you calculate what is called a classifier, and that's 3 parameters that are associated to each measured current distance trace relative to what we call a reference vector R. And this reference vector is, for these experiments that I'm going to show you in a second, the reference vector is simply an exponential decay, because that's the sort of event-free current distance curve that we're expecting. Yeah, and then we basically compare every measured current distance data to that reference, right? The first parameter to do that is the so-called vector distance. Yeah, so the Euclidean distance in vector space between the measured current values and the reference vector. The second parameter is the angle between, in this case, the current vector minus the reference vector and the reference vector. Again, bear with me, I'll illustrate what that all does in a second. And then the third parameter is what we call the reduced Hamming distance. Now, the Hamming distance is normally, well, it's a way to compare especially binary vectors, and it's the relative number of component exchanges that renders 2 vectors identical, according to the definition. The way we use it here is basically to arrive at a measure how many data points of the current values that we measure lie below the reference vector, right? So it's essentially a measure for the positive and negative deviation. And with each parameter, we've got 3 parameters for each measured trace, right? We can then start to compare different traces, but now in 3-dimensional space rather than 2000-dimensional space. Now, what does this all mean? Okay, so let's have a look at a toy example. The reference vector is shown here. That's just the black curve, an exponential decay of the current distance curve. Then I've constructed two example traces. The red trace here shows a plateau, or first an exponential decay, then a plateau, and then a break off, right? And then we've got the blue trace, which shows the same plateau height, but a shorter break off distance. And if you calculate the vector distance following according to the formula I've shown you on the previous slide, delta x, we get 0.56 for the red trace and 0.32 for the blue trace. And in a sense, this is already dimensionality reduction, if you like, because you can use this single number to differentiate the blue from the red curve, right? So now, of course, in reality, it'd be nice if it would be that easy, but you can see here what happens in the second example, where we now compare the blue and the green trace. The blue trace has got a higher plateau and a shorter cutoff. The green trace has a lower plateau and a longer cutoff. And as you might expect, the vector distance to the reference vector is actually quite similar, 0.33 and 0.32. Right, so that's not good to differentiate these two traces. Now, what about the vector angle? Again, the green gives us 122, the blue 126, some difference, but not great. And then finally, the reduced Hamming distance, and this is now working a little bit better here. 24% of the traces have different sign, or are below, basically are below the reference vector, whereas for the blue one, it's only 15% of all components. And then, you pull all these three parameters together to produce a point in three-dimensional space that's defined by the individual parameter values. And the idea again would be that if these points in three-dimensional space are close together, then the original data traces are similar. If the three-dimensional data points are far away from each other, then the original data traces are dissimilar. Now, applying this to an experimental system, OPE, or oligophenylene ethynylene, this is sort of a fruit fly molecule, is very well studied in the field, so a good way to test a method, right? This is the molecular structure here. It binds to gold on this side and that side, right? And then basically we study charge transport through this structure. Right, and again, as I said, the conductance is extremely well characterized and so on. Now, when we perform these experiments, then we'd record, let's say, 2,000 traces. Again, every trace has got about 2,000 data points. And when we now convert this to this reduced dimensional MPVC space, then at first we see the scatter plot on the top left-hand side, and you can kind of see that, well, there's a cluster up here, and there's a cluster perhaps up here, and maybe something around here. And I'll come back to that in a second. Now, if you analyze this data set in the conventional way with a 1D histogram, you actually don't see anything really, right? So there's no clear molecular distinct feature here, which would allow us to extract the molecular conductance. Partly because there's a lot of empty exponential traces in this data set. On the right-hand side here, you see sort of a two-dimensional histogram, mostly exponential decays. Notice this is a logarithmic scale there. A very faint plateau, right? So you can see there is something, but it's really difficult to pull this out. Then we cluster the data based on the assumption that there are three clusters. In this case, we used Gustavsen-Kessel fuzzy clustering, but it's not the only method you can use, of course. Now, that then produces three clusters, as you put in. Now, we've got three colors here. The blue one, that's the cluster that's at the origin, right here, it's about. And it turns out these are mostly exponential decay, so empty traces, if you like. The blue one, sorry, the red one, that's the one down here. Now you actually see a distinct molecular peak that corresponds to the molecular conductance. Yeah, and you can extract from the position of this peak, you can actually extract the current that's passed through the molecule by analyzing the 2D histogram of this data set. We also now find a very distinct plateau shape. Right? There's also still some exponential decays here, so that the separation isn't perfect, but the signal to noise ratio is much improved. Now, surprisingly, when we looked at the green cluster, we actually found that there's also a peak. Yeah, so maybe it's a bit squashed against the left-hand side, but there's a clear peak that corresponds to something like molecular behavior. Now that we didn't expect, and we didn't know what that was until we sort of read the literature properly, right? And went back to the literature. It turns out that this had actually been observed before, right? Now, Wu et al. in a paper in Nature Nanotechnology some time ago measured a conductance of 0.47 nanosiemens, whereas for our data we get 0.28. That's actually fairly close. And so what does that all mean? Well, again, the main molecular events are in the red cluster, right? You could see that there was something there, but it was actually much more easily identified using MPVC-based analysis and clustering. The green subpopulation was of low abundance and does not show at all in the all data representation. And that's something we would clearly have missed, right? Especially because we didn't know that something like that would exist. But if we then compare to previous literature, the physical interpretation of this very likely involves biomolecular junctions. And again, unexpected by us, but observed before, right? The data, the previous data suggests that this might be a situation like that. So these are the two electrodes in yellow. As you pull them apart, you have interaction between two molecules here and corresponding a lower conductance because it's just not such a good molecular wire, right? So again, that shows you how you could use a dimensionality reduction approach to identify a population in the data that you weren't expecting, but in this case, what had been observed before. Now, in the second example, I'm showing you a case where the data, well, where this type of analysis provides or identifies not only unexpected, but a previously unknown subpopulation in data. And this is basically using a different approach. It's using an unsupervised data classification approach using a combination of image recognition and dimensionality reduction. Now, and this goes back to the idea of Anton Vladica when he was a postdoc in my group. I should say that all the experimental data that I'm going to show in the remainder of this talk were measured by Anton when he was part of Michel Callamy's group in Switzerland, but we then reanalyzed the data using this new method while he spent time with me. Now, again, when you look at the data, again, this is the labels are missing here, but you basically have a current as a function of distance or conductance as a function of distance trace. And what you see here is an example of one of those traces that Anton measured. And you can see here these sort of plateau features and sort of by and large a wiggly line. But what we thought was, well, normally we think of this data as single or one-dimensional data, but basically we look at them as images, right? So these are black and white, a simple image, right? And the question we asked was, well, can we use this as image data and extract salient features from that? Because image recognition is, of course, something that's very well established in machine learning and then AI, especially deep learning. Right, and there are, of course, already image recognition networks available. Networks like AlexNet, ResNet, or VGGNet, for example, they are open source, right? So you can access them. They're already trained on millions and millions of images and photos, which, mind you, were initially hand labeled, but of cats and dogs and ships and so on. And so this is how these networks are trained to then recognize unseen images. Yeah, so once they are trained with this large amount of data, they can then identify cats and dogs and ships that they hadn't previously seen. And we thought, okay, well, they certainly haven't seen our conductance distance traces, so they are, not domain specific to what they have been trained on, right? But can they still extract features from these traces and identify, some meaningful differentiators when we compare different molecules, for example. And we've the network that worked best for us is AlexNet, right? And again, I'm not going to go in detail here about the design of AlexNet. Again, this is available if you're interested. But basically, the idea is that it uses images as input data, right? Yeah, so for example, this photo of a dog would use that as input with a certain resolution, right? And it would have, it's typically, well, it's composed of a feature extractor, which tries to extract the salient feature in what identifies this as a dog, for example, then it passes this on to a classifier, and that classifier is ultimately responsible for specifying what's seen on the input image. Now, what we've done in our case is different. Well, we've replaced the classifier at the end here with a dimensionality reduction technique, and in particular an autoencoder. Now, what that autoencoder does, it basically uses the output of the feature extractor, then reduces that down to a very small number of dimensions, even as small as 2. And then, well, the autoencoder does try to then reconstruct the original data set, but that we're not interested in. We just extract the numbers at this stage here and use that for our clustering plot, right? So our input now are the conductance distance traces. They feed through the feature extractor. And then we use dimensionality reduction to, because there are thousands of individual features, 9,216 in the case of AlexNet, right? We want to boil this down to a small number that really differentiate the data that we're interested in. And Anton studied sort of these 3 molecules here. There are phenylene diisocyanide molecules, which bind to 2 electrodes via the isocyanide groups here and there. And in this case, they had 3 different analogs. One where this residue, this R here is just hydrogen, and they call for some reason call that BDNC. In one case, it's methyl, that's MBDNC, and then there's tert-butyl, which is TBU-BDNC. Right, and that kind of allows you to compare the effect of structure on what you measure, and so on. Now, if you just compare BDNC and MBDNC, the one with the hydrogen on and the methyl group on, right, you clearly see that for example, the MBDNC starts from the contact up here, then you pull the electrodes apart, you get this plateau feature, and then eventually that plateau breaks down and goes to basically 0 current down here. You can extract the molecular conductance. You've seen this plot before. Right. On the other hand, BDNC in this case actually produces 2 plateaus, and the second plateau at a larger distance. The idea here is that you actually start to concatenate molecules, which is easier with the BDNC and not so easy with the MBDNC, right? And therefore you see distinctly different behavior for these 2 cases. Now, the first thing to do was to apply dimensionality reduction directly to the conductance distance data. Right, and again, there are a number of techniques that we've used here, BDNC in blue, MBDNC in orange, and tert-butyl BDNC in green. All 4 techniques that we used here, PCA, MPVC, TSNE, and an autoencoder, work reasonably well in the sense that you have different molecules in different regions of this plot. But they're not great, of course. The separation isn't fantastic. There's a lot of overlap between these traces. When you then look into with each molecule separately, because of course they were measured separately, then it also looks like none of these molecules actually produce any subgroups, subpopulations. And based on that, you would think each molecule represents a single ensemble. Now let's see what happens when we use an autoencoder for dimensionality reduction, but bolted onto the feature extractor output of AlexNet. Now, at first, it looks very similar, right? So you can see here, now helpfully color-coded in a different way, but black, red, and green, right, are BDNC, MBDNC, and tert-butyl BDNC. There's not great separation between these molecules, at least some overlap here and here. But if you then, when you then look into each individual molecule, you all of a sudden start to see something different. Namely, for MBDNC, for example, we see a larger cluster, cluster 0 in red, and we see a smaller cluster, a subpopulation here in brown, right? And when we then analyze them separately, we find that, let's say for cluster 0, which is the red one, we actually have a slightly higher plateau height, and we have a significantly less well-defined breakoff in this region here. Whereas for cluster 1, the plateau is a bit lower and the breakoff is a lot cleaner. It's a lot sharper, as it basically should be. And it turns out that the plateau height between here and here differs by pretty much a factor of 2. Yeah. Now, what does that all mean? First of all, we see 2 distinct subpopulations in this data set that hadn't been observed before. to establish the physical meaning is of course difficult. It's not a small task, but at least now we have the statistical tools to do that. Right? Now, the tentative explanation that we've come up with is that because the plateau height is larger by a factor of 2 in cluster 0, and that we get a steeper, cleaner break-off in cluster 1, is that perhaps cluster 1 has proper single molecule events, where you have an individual molecule bridging the gap and then breaking, whereas in cluster 0, you may have 2 molecules or small aggregates bridging the junction before it finally ruptures. Yeah, but you can actually now start to interpret these data in much more detail than you would be able to with conventional methods. Now, some conclusions to this point. Charge transport data for these 3 molecules were reanalyzed. We found subpopulations for MBDNC, but also for TBDNC, which I haven't shown here in the interest of time. When we combined image recognition for feature extraction with dimensionality reduction, we did not see subpopulations for BDNC. This observation was neither expected or previously known for these 2 molecules, right? And of course, as I said, the physical interpretation is tentative. The network has no domain-specific knowledge, and it does not have to, right? The feature extractor was trained with unrelated data, but the feature extraction was nevertheless successful. And that, of course, has implications, because now, well, there's this notion that if you want to use deep learning, you need a large number of training data, but that's really not the case necessarily, right? Because we can sometimes use unrelated data to train the network and then use a small amount of domain-specific data to then actually arrive at an analytical solution, to an analytical result. Yeah. And then perhaps an ultra brief outlook, right? I've talked a lot about what a molecular event might be like, and whether they have different shapes and composition, temporal bursts, and so on. But my personal interest is beginning to shift, from what is a molecular event to what is not background, right? And what is not background signal. And that takes you into the area of anomaly detection, for example, which is, of course, highly relevant in other areas like network security and intrusion, particle physics, medical imaging, and other areas. And I'm really interested to see how that can actually be integrated and whether there's something we can learn from that for our data. But with that, I'll stop and thank you for your attention.